Hello, in this video I will be talking about Polycomb group proteins. They were first identified in Drosophila as regulators of homeotic genes. Homeotic genes are genes that regulate the development of anatomical structures in various organisms, including insects, plants, and mammals. Polycomb group proteins are associated with transcription repression. It is due to repressive marks that lead to the formation of facultative heterochromatin. Unlike constitutive heterochromatin, facultative heterochromatin has the potential for gene expression at some points of development. These proteins are also highly enriched on inactive X chromosome, and they maintain embryonic stem cells in an undifferentiated state by preventing the transcription of genes encoding proteins such as transcription factors that are required for differentiation into different cell types. PREs, or Polycomb response elements, are DNA regions that recruit Polycomb group proteins. They may be located both upstream and downstream of the promoter. Interestingly, it was shown that even in the presence of Polycomb group proteins bound to PREs, transcription factors bind to the promoter. However, RNA polymerase II is not able to initiate transcription. The exact mechanism of this interference with transcription initiation is not known yet. Polycomb group proteins are organized into two Polycomb repressive complexes, PRC1 and PRC2 respectively. PRC2 consists of 4 core subunits. PRC2 complexes may also have additional proteins that modulate their activity and play a role in targeting PRC2 to chromatin. One of the PRC2 subunits, specifically enhancer Z homolog, contains Z domain. Z domain has a histone methyltransferase activity. Enhancer Z homolog is able to mono-di and trimethylate lysine residues on N-terminal tails of histones. Different states of methylation have different biological roles. Even though trimethylated lysine 27 is linked to gene repression, it was shown that monomethylated lysine is associated with gene activation. Dimethylated form is prevalent in the genome. However, little is known about its function. Another PRC2 subunit, specifically EED or embryonic ectoderm development, binds to its own mark as a trimethylated lysine 27, and it provides a positive feedback mechanism that is important for cellular memory and inheritance of this mark through cell division. PRC1 is also a multiprotein complex. Its RIN subunit is able to monoubiquitinate lysine 118 on histone H2A. Another subunit, RIP, binds to this mark, and it also provides a positive feedback mechanism. PRC1 also contains chromodomain CBX that reads trimethylated lysine 27. This led to the development of a hierarchical recruitment model, whereby the trimethylated lysine 27 on histone H3 placed by PRC2 is read by PRC1. However, it is not always the case, and the reverse mechanism is also possible, whereby PRC1 is recruited first, followed by PRC2. In this model, the monoubiquitinated lysine placed by PRC1 complex is recognized by PRC2. The exact mechanism of this binding is not known. PRC2 complexes, besides direct methylation of lysine residues, may also recruit some additional proteins, such as histone deacetylase complexes, that lead to the removal of acetyl group and are associated with silence genes. PRC2 complexes also recruit DNA methyltransferases that provides a link between histone methylation and DNA methylation.